Nearly every group of friends has at least one friend that loves to be the life of the party. Often, they facilitate a network of communication and help those more introverted individuals transition to a slightly different version of themselves, more appropriate for the context. RNA binding proteins are just like these lively friends of ours. They are a diverse pool of proteins that pair with different friends, or mRNAs, and aid them in a transition to a slightly different state, or isoform. Once they've spent time with one friend, they move on to the next, and before you know it, these RNA binding proteins have helped to regulate an entire network of mRNA friends, and now the party has started. And these RNA binding proteins are crucial during human development, just as these lively friends of ours are crucial during party development. But what happens when these RNA binding proteins go awry? How will this network of communication and necessary transitions now be facilitated? Unfortunately, we don't have to go far to find the answer, because that's precisely what happens in myotonic dystrophy type 1, the most common form of adult muscular dystrophy. A prominent feature in this multi-system illness is significant muscle wasting, yet the cause of this wasting remains unknown. That is exactly what our lab is trying to figure out. And my project studies the role of one of these crucial party-regulating friends of ours, an RNA binding protein called SELF1. Normally, SELF1 levels are high at birth and decrease with age. In myotonic dystrophy, these levels become elevated. And while we now know that elevated levels of SELF1 lead to muscle wasting, we don't clearly understand how. One thing we do know is that this is a very complex protein, and it functions in two different areas of the cell, the nucleus and the cytoplasm. In these two compartments, SELF1 regulates many different RNA targets, or friends, and very different processes. So I wanted to figure out which roles of this protein lead to disease. I'm answering this question by studying the RNA binding protein in different contexts. Using animal models, I have overexpressed SELF1, either primarily in the nucleus or the cytoplasm of muscle cells. And what my work has found so far is that overexpression of this protein in just the nucleus leads to muscle wasting, yet overexpression in the cytoplasm does not. And this result is so exciting because it narrows down our list of mRNA targets. And when we understand which of these transitions are necessary and symptom-causing, we can better develop therapeutics to fix them. I aim to discover those crucial mRNA transitions that are regulated by this RNA binding protein and necessary for proper muscle development and function, not only in myotonic dystrophy, but in each and every one of us. Thank you.